Hello, everyone. My name is Jasem Alkazi. I am a student at the University of Arizona, and today I'm going to be going over the Red Onion Osmosis Lab. We're specifically going to be looking at red onion cells and how they behave under different environments of different solutions. And we're going to be talking about what those solutions are later on. But before we get started, I would like whoever is leading this session to quickly take a moment and pause this video and have you guys all introduce yourselves to each other because given this current situation of the pandemic, I think a lot of us haven't had enough student interaction with each other or peer interaction. So go ahead, pause this video and introduce yourselves to each other. Okay, so hopefully you got to know a couple of your peers. We can go ahead and get started now. So for today's lab experiment, we are going to be needing some materials, specifically a microscope because to the naked eye, we can't actually see what would happen if we put different solutions onto a red onion layer. We won't be able to tell what's going on to that red onion layer. So we actually want to use a microscope because on a microscopic level, we can actually see what is going on once you add these different solutions and you build this different environment for the onion cells. Obviously, with that, you also want an onion, specifically a red onion, because they have this hyperpigmented layer, or or hyperpigmented. I meant to say pigmented layer. Pigment means coloration, so they're highly pigmented, and they have this dark color that allows us to see what's going on when you put these solutions onto the layer of the onion. You're also going to be needing a slide. A slide is something you put your onion layer on, or whatever it is you're experimenting on. You put that under the microscope, and then you put a cover slip on top of that slide, and that protects our microscope from getting damaged when you zoom in to whatever it is you're looking into. It prevents the microscope lens from actually hitting whatever solution we're looking into. And then you're going to obviously be needing your solution. So we are first going to be using distilled water, which is one of our environments we're going to be looking at, and 15% salt solution. Um, and then we can look into some background info real quick. So what is osmosis? Osmosis is the movement of water through a selectively permeable membrane. Water moves from a low concentration of solutes to a high concentration of solutes. Another way I think about it is water moving from a region of high concentration of water to a region of low concentration of water. So for the remainder of our lab, that's how I will be referring to it as. And I promise by the end of this, you're gonna hear me say it so many times that it's gonna be stuck in your head. But we will also be using 15% salt solution. And this solution has more solutes than the inside of the cell and is hypertonic. You might be wondering, well, what is hypertonic? Don't worry about it just yet. We're going to be getting to that later on in the lab, but perhaps take that word and try to piece it together. What does hyper mean? What does tonic mean? And maybe come up with an idea as to what it actually means. And then also we are going to be using distilled water, which is hypotonic when compared to the cell. And again, you're going to be learning about what that actually means later on. Before we move on though, I want you to, again, pause this video once I'm done. Pause this video and think about where osmosis takes place in the body. So there's even a hint for you guys in this slide. So take a couple of moments, go back to the peers you talked to at the beginning of the session and ask them or talk to them, where do you think osmosis takes place in our body? So hopefully you got to talk to your peers. Osmosis actually takes place in the kidneys. So you know all that weight or all those liquids and drinks and all that stuff we drink in or we take in throughout our day. Well, guess what? The kidneys are the things that filter all that waste. And how do they filter all that waste? They do it via osmosis, and then they filter it out through urination, aka pee. So here, that's another, physiological thing for you guys to kind of just, have at the back of your head if you're ever wondering where osmosis takes place in the body. Okay, so now we can move on to an analogy that I think of when I want to maybe remember osmosis in a different way. So consider a room, or consider you with a couple of your friends in this area where you have a semi-permeable membrane as a door. Outside this room, there is a bunch of students, a lot of students, kids specifically. And on the inside of the room, there's very little kids. Now, the kids outside the room, they don't have any candy or anything. They're just, just there. Maybe they have like a couple of candies to share between them, but for the most part, they don't have too much. Whereas the kids on the inside of the room, they have a lot of candy and there's very little of them. Now, given what we talked about osmosis and how water travels from a region of high concentration to a region of low concentration, and in this case, the kids being the water and the candy being the solute and the door being the semi-permeable membrane, where do you think the kids are going to be traveling? Take a moment and discuss. So hopefully you discuss this. So like we said, there's a lot of kids outside the room. These kids will want to open that door, go past that door, and go to where there's this little amount of kids, but this huge amount of candy, right? So they want to go from a region of high concentration of kids, solvent, aka water, to a region of low concentration of water, aka kids, right? And to the solutes. So hopefully that made sense to you guys. That's a helpful analogy. Sometimes I think about if I'm ever confused about osmosis. Again, if this still isn't clicking, don't worry, we're going to be looking at the microscopic implications of this as well. So hopefully it will make sense then. Before we do move on though, here's a quick procedure as to what you're going to be doing in today's lab. So you're going to be having a demonstrator cut the red onion into 1 centimeter slices. And then you're going to be using forceps to remove the thin onion skin, and then place it on a clean microscope slide, the one I showed you at the beginning of the lecture. We're going to cover the onion skin with a slip, and we're going to repeat this if you decide to. But also, we're not going to be removing the cover slip when using a pipette to drop a single drop of distilled water. So what you're going to do is you're going to take your pipette, you're going to suck up some of the water that's laid out for you, and then drop that little drop of distilled water on the corner of your slide or your slip, and you're going to be you'll see that the water gets sucked in by the slide, and there's a demonstration coming up ahead, or a picture that will show you how that takes place. And then what you can do is you can do one of two things here. You can either continue with this same slide and now take 2.5% saline solution, or 15% saline solution, that should say 15% saline solution, apologies for that, and put that on the corner of the side itself. And see how the water gets sucked in once again. Once you're done with that, you can look at it under the microscope. So what I recommend doing before you guys go ahead and start with this is first cut your onion layer up, take out the layer which is highly pigmented, and place it under, or place it onto the cover slide, and then put a paper slip on it. And then look at it under a microscope, find a good region where you can see a lot of the cells, and then get a good picture, and then add your distilled water. Once you add your distilled water, look under the microscope. This process may take a while, so don't rush it. Let the distilled water get sucked in and see how your onion cells behave. Once you're done with that, like I said, there are two options. The first one is you can take the 15% salt solution and add it to the corner of the slip and see how that affects the red onion cells. Or what you can do is you can go ahead and create a new slide with a new onion layer and just put the 15% salt solution. For our purposes, we're going to be using we're going to be continuing on with our distilled solution. And you'll see why we're going to be doing that in just a moment. But just one more time, if you are having trouble visualizing this, you cut your red onion, and then you peel a thin red layer, or you remove the inner layer that is highly pigmented first, and then you peel a thin red outer layer with a tweezer or your fingernails in this case, and then you spread it out on the slide. And then you can use your microscope to look at it under the microscope itself. And then here's what I was talking about, where you put the water onto the corner of the slide and the water kind of gets sucked in, as you can see in this third image here. Hopefully you saw my mouse hovering over that. And once you're done with that, you want to make sure your microscope is set up correctly. So have it be so that the light is bright enough for you to actually look at the cells, not too bright so you don't blind yourself. So just be careful with that, and have it be positioned so that you can see a lot of the cells. And as you can see, our experimenter didn't even have that much of a layer from our onion. So it's okay if you don't get too much layer, as long as it's pigmented and you can see the cells, you should be good to go. And now we have a short video for you guys to kind of watch. And we're not going to be watching the entire thing in slow motion. As I said, this process does take a while. So we're going to go through it whilst, talking about what's going on in our solution. So here, as you can see, the cells are normal in size. You can see what's called a cell wall here. And you can see the inside and the outside, and you can see, the onion cells are normal in size, nothing too crazy. This is called isotonic, which we'll talk about more later on. But there's essentially no net flow of water. There's no movement of water from either inside the cell or outside the cell. It's just, in a sense, it's at an equilibrium. There you go. Students, some university students can pronounce that. But if we go ahead into the video, the scientist or the student now has put in the distilled water. So as you will see, with the distilled water being put in, the cells kind of shrivel up. And again, this process takes a long time, sometimes around 10 minutes in some cases. This video is 20 minutes long. I'm just going through it really fast, so it might take a while. So as you can see, the water is leaving the cells. So as you can see, these cells got shriveled up. They were first, or prior to this, they were all, normal. As you can see, they shrink in size, they get all shriveled up. What does this imply? Maybe pause the video and take a moment. Explain to me what you think is going on. What's going on here? So hopefully you pause the video. Like I said, water is leaving the cell, right? So there is a higher concentration inside than outside. So water wants to leave the higher concentration. It goes from a region of high concentration of water to a region of low concentration of water. So it leaves the cell, keep allowing it to become deformed like that. And then if we keep going, you can see a lot of the cells undergo the same process. And as you can see, the cell wall kind of stays intact. It doesn't actually shrink. It's these inner layers that actually shrink, the plasma membrane and all. And then now we're going to be actually just seeing what happens. As you can see, it's still shrunk as we progress. Now here, actually, the student adds the 15% salt solution. And as you can see, slowly but surely, actually, I don't think they've added it just yet. There we go. It looks like they added it just then. They've added the 15% salt solution onto the distilled water. And it looks like they had a slight error where the sides might have moved. Apologies. The slide kind of moved, but if you can just recalibrate and fix your slide under the microscope, you should be good. But now essentially what you can see is these shriveled cells kind of go back and normalize in size. Look at this one. Look at up top. You can see that, you know, they kind of normalize. Look at this one on the right, on the very right. Look at how it becomes normal back in size. And also look at these round ones. So maybe explain to me what's going on to these round ones, what's going on here, which this is why we chose to continue on the distilled water. So take a moment and tell me what's going on. What do you think is happening? So essentially what's going on for these cells is water is going back into the cells, right? So a region of high concentration of water to a region of low concentration of water. So once we added the distilled water, now we had a region of high concentration of water outside the cell. So now when we added the 15% salt solution, that water wants to go back into the cell, right? It wants to go back into the cell. So now you have this normalizing cell, which is an isotonic solution. But what happens when these cells get too much water going inside of them? What happens is that they become this round shape and there's a lot of water going inside. It's rushing inside the cell because of the whole high region of high concentration of water outside versus inside. So take a moment, let this all get in, maybe watch the PowerPoints are provided to you, maybe watch this again and try to explain in your own words, maybe using the other definition we talked about earlier to explain what's going on here. And then we can go ahead and look at plant versus animal cells. So we talked about the cell wall very briefly. I want you to look at these two pictures and let your instructor know what are two differences you see in the animal versus the plant cells. They're also highlighted for you as a hint once again. So as you can see, with the plant cells, you have something called a cell wall. It gives protection to the cell layers, preventing it from collapsing under osmotic pressure. And we also have the chloroplast. So like we said, cell wall gives a structure, but what about the chloroplast? So the chloroplast is, as you may recall, is where photosynthesis actually takes place. So chloroplasts absorb sunlight and use it in conjunction with, you know, water and carbon dioxide gas to produce food for the plant, right? Chloroplasts capture the light energy from the sun, produce a lot of things, and you have this photosynthesis. And then once again, cell wall provides that protection, gives it this rigidity. Sorry, I've been hovering over at the bottom, but the cell wall gives this rigidity. And then for the animal cells, we have the centrioles, which we're not going to focus on today, but they have that, which is somewhat different than the plant cells. And then moving on from that, we can look into what's actually going on. So the types of solution we talked about were isotonic, hypertonic, and hypotonic. Iso means same. So remember how I told you what iso means, tonic means that might help. So iso means same and tonic means strength. So there's the same concentration of solutes and solvents. So we don't see any change, right? There's no net flow of water anywhere. Whereas hypertonic, there's more, and tonic means strength, and there's more solutes and less solvent, right? So, you know, water is initially inside, then there's a higher concentration inside. So the water leaves from the higher concentration to a lower concentration. Hypo means less and tonic means strength. So fewer solutes and more solvent. So now, as you can see, the water now that was outside, so in higher concentration comes back in, and now your cells become this round shape and they become this hypotonic solution. As you can see, some normalize, some become very hypotonic, but one thing to notice once again is the cell wall remains the same because it provides that rigidity and protection for the cell itself. Now what I would like for your professor to do is break you out into breakout rooms, break out into groups of 3 and describe what you see in pictures. Be creative and think outside the box. Describe what you think might be the reason for the phenomenon by sharing your thoughts with your peers. your instructors will be walking around to ask questions and ask what you're thinking. Maybe also come up with a hypothesis as to, what's happening. The hypothesis doesn't even have to be like a proper hypothesis. It could just be like a normal question, or, just a normal something like a blurb, what you think is happening. And then after that, we can also discuss it after pairing with more groups. Once you pair with more groups, with other groups, discuss your ideas with them as well. Collaboration is key for experiments like this. So go ahead, and once you're done with your own process, so once you're done with your own um, model of explaining what's going on, run it through other peers, explain what's going on to them, and share your thoughts, share your ideas. And then what we can do, or actually before we do move on, we're also going to be choosing for this demonstration a couple of people to share your models. And no pressure, it can be anything. Like I said, there is no right or wrong answer for this. We want you to volunteer, we want you to participate. The goal of today's lab is to have you guys get engaged and get interested in biology and how stuff happens in these microscopic levels. Finally, summarize the lab and explain your result if you can, and talk about your hypothesis. Do you support it? Do you reject it based off of what you talked about with your fellow peers and what you talked about with your instructors who were walking around with you? And then one question I want to leave you guys with before we end this is, how did the 15% salt solution affect the structure of the onion cell? And give a reason for why this occurred. So as you saw in the video, the scientists actually just put in the distilled water and then the 15% salt solution, correct? What would have changed if you had just put in the 15% salt solution into a normally peeled onion layer? Under microscope, what would you have seen? Given the phenomenons and the phenomena and changes we saw, what do you think would have happened? Where would have water traveled? Think about, high region of high concentration of water to a region of low concentration of water. And I will let you go with that. Thank you for your time. Thank you for having me. And if you're seeing this video, it means you're somewhat interested in biology. If you're interested in learning more, I highly recommend that you guys go out to your nearest library and rent a microscope and run this experiment at home carefully, obviously. Run it with different pigmented vegetables or fruits, and see how these different solutions affect them. And if you're really interested, please reach out to your teachers or the University of Arizona, and we would love to make more of these for you guys. Our goal is to have you become interested in science and in biology. So if you have any questions ever, feel free to reach out to your professors or your teachers. And once again, thank you very much for letting me go on for so long. Bye.